So today's lecture is about cell and tissue. This is the second day of our 4 class for anatomy. So this is just a reminder. I believe you know all about the structure of cells in your biology lecture. But this is a cell. A cell is surrounded by the cell membrane or plasma membrane. Inside the plasma membrane, we do have cytoplasm, which is called cytosol. And inside the cytosol, we have multiple organelles such as mitochondria, endoplasmic reticulum, ribosomes, nucleus, and rough endoplasmic reticulum, smooth endoplasmic reticulum. So this is a cell. So who start to notice a cell? So we have to give some credit to Robert Hooke. Robert Hooke, he's the little guy from small little island, which is to the west of the main British Isles, that's called Isle of Wight. So he started looking with the microscope and start seeing small little holes. And these small little holes, like he gave it a name called cells because they look similar to the small little chambers or little rooms or prison cells, in the monastery, cells. That's why he gave the name cells. With the advancement of the microscope, these German scientists invent more and better microscope, and then these can start seeing, and our understanding of cells is much higher and higher. Of course, a human being is made of trillions of cells. So the cell is the basic functional unit. So if we have time and if we put these one cell, another cell, another cell, another cell together, and when it comes to like trillions, then we get a human beings. But to make it simple, this is one cell, another cell, another cell. Inside the cell, this is the cell membrane, which is the boundary of one cell. Inside the cell, inside the space is called intracellular space. Outside of the cell is called extracellular space. There will be small little spaces in between each and every cells, like here and there. These are called interstitial spaces. Of course, for us to see the cell, we have to use the microscope. So we have to prepare the tissue, look under the microscope, which is something like histology, which is a microscopic structure of the tissue. So a cell is the basic functional unit of livings. If we combine cell with the same function, we get a tissue. So tissue is the group of cells. A cell is the basic structural unit. And there are 4 types of tissues in our body. They are epithelial cell, connective tissue, muscle, and nervous tissue. So a human being, you and me, whether you are black or white, tall or short, doesn't really matter. Any human beings, we are made of only 4 types of tissue, which are epithelial, connective, muscle, and nervous tissue. So yes, I believe we all remember cells already, and I'm not going to explain a lot about the cell and its contents and plasma membrane function. And this diagram is a very nice diagram, which is showing the plasma membrane, which is made of phospholipid bilayers. And inside the lipid fat, there will be some protein integrated into the fat. We call this type of protein is called integral protein. And there will be proteins which is just attached on the outer surface of the phospholipid bilayer. And that's what we call peripheral proteins. So plasma membrane is the boundary between the inside of the cell and the outside of the cell. Cytoplasm, it is just like a jelly-like inside the cytoplasm, inside the cell membrane, it is called cytoplasm. A cytoplasm is filled with the cytosol, which is jelly-like fluid, within which all our mitochondria and nucleus, they float inside the cytosol. Content of the cytoplasm, I believe that's what we study during the biology class. So let's skip and then try to go and study some anatomy. So as I always say, a human being is living, which is made of body and mind. So everything we see, we try to understand and clarify and classify everything we see. A body can be classified according to their age, according to their sex, and according to their skin color. If this is a white person, this is a black person, this is a colored person, or according to their sex, you know, like male or female, or according to their age. Hey, little one just born, newborn, neonate. So neonate is age from birth to 1 month. And infant is from 1 month to 1 year. And after 1 year, until it comes to puberty, that is called child. And from puberty, let's say teenage, that is young adult. And adult mean like you and me, might be 18 or 21. And old age is difficult to define what is old age because humans are living longer and longer. But I would say, you know, retirement age. Like previously, it was 60 as a retirement, and now it has changed to 65 or 67 in some other countries. So this might be the old age. I cannot definitely say it is a little bit, it could be a little bit controversial, but that's the way we define, we classify a human being, human body into different classification. Of course, mind, we will not be going to attempt, but depending on their personality, you know, mature personality or immature personality. Let's leave it for the psychologists and psychiatrists. So we know that our human body is made of 4 types of tissue, and these tissues are made of cells. To see the cells and tissue, we have to look under the microscope. And let's start with this. So now if I give you a slide and you start looking at that and you see under your microscope, low power microscope, as you see something like that, which is a tubular structure with a central lumen in it. And you can see one group of cells, which is one tissue in a very different color, like dark purple color, and which is surrounded by another layer of tissue, which is pink color, and the outermost is slightly different as well. So let's go and zoom in into this area, and that's what we see. This is the luminal layer, and these are the cells we can see. We can see very deep multiple purple. Why is that? Because that is the dye, you know, stain taken by the nucleus, which is giving as a high color, deep purple color nucleus. And it is sitting on another type of tissue. Let's keep looking, And now we can see the lumen. All the cells are projecting into the lumen and they are sitting on another type of tissue. And this is a very good microscopic slide. You know, if you look under the microscope and then you start seeing, hey, this is the lumen. The cells are projecting into the lumen. And these cells are sitting on another type of cell. And there is another type of cell here. So at least in this microscopic slide, we can see 3 different types of cell, one here, one there, and another one here. Just keep looking at similar and similar. What is the importance of these slides? Of course, they have multiple cells. Why do we say multiple cells? Because we can see multiple nucleus. One nucleus, one cell. One nucleus, one cell. Multiple nucleus, multiple cells. So these cells are so much cellular, a lot of cells. It is quite different from another tissue layer here. So now it is just like, you know, just looking from the very high bird eyes point of view, and it's just looking at like that to the San Francisco Bay Area. But let's say now if we just zoom into that area and we start seeing the luminous structure here, and these are the long tall cells we can see. Yes, so this is the epithelial tissue. So epithelia, the characteristics of the epithelial tissue is number one, cellularity. As we see, there are many cells, loads and loads of cells, and they are sitting on one type of tissue, which is the connective tissue. And polarity, they are exposed to the external environment. That is what we call polarity. So that is a very important characteristic features of microscopic features of epithelial tissue. And each and every cells, they are contacting to each other with a specialized connection. They do not have direct blood vessel, therefore they are avascular, but they do have direct nerve supply, and therefore they are innervated, and they have a good healing potential, and therefore they have good regeneration power. So these are the characteristics of the epithelial tissue. So if we look, this is the diagram of the epithelial tissue, one cell here, another cell here, another cell here, combination of cell become tissue. Epithelial cell, they are exposed to the external environment, which is a luminous surface, and they are sitting on another type of cell, which is the basement membrane. So that is what we call polarity. One side is exposed to the environment, other side is sitting on the another type of tissue. And if you look at that, this is the nerve, which is colored with the yellow. They go in between the cell, but this is the red color, which is the blood vessel, capillary. They do not go directly into the cell. They just give nutrition to these cells through diffusion. So in between each and every cells, we can see the desmosome, we can see the tight junctions, we can see the gap junctions. Tight junction means they are really tight and nothing can go across this part of the intercellular space. But there is a gap junction, which means there will be transfer of signals or substances across this gap junction in between cells. So yes, that is a much bigger diagram. So how do we classify cells? Of course, we have classified everything we see here. We can classify epithelial tissue into according to their cell shape and according to how many layers of cells they have. So if the cell is flat out, then that is what we call squamous cell. Square, box, cuboidal shape, cuboidal cell. Tall like a columns, pointing, projecting, columnar cells. If this squamous cell is only one layer, we will say simple squamous epithelium. If the squamous cell is multiple layer, we will say stratified squamous epithelium. The same thing like, simple cuboidal or stratified cuboidal, simple columnar, stratified columnar. And pseudo-stratified, pseudo means it's a fake, not real. Pseudo-stratified means it looks like it is multiple layer, but actually it is only one layer. That is what we call pseudo-stratified epithelium. So pseudo-stratified columnar means it looks like multiple layer of columnar cells, but they are only one layer. Transitional, it is more like a combination of all these. So if the cell shape change throughout the layer, and what happened is it is very useful for the different function. It takes all the good things about different cells. That is what we call transitional epithelium, which we use it for the urinary bladder. So which can be expanded, get bigger, and after voiding, the urinary bladder becomes small. So in this type of cell tissue, we are going to use the transitional epithelium. So basically it is squamous flat cells like the scales from the fish, like a cuboidal cell, and tall columns like a columnar epithelium. So now look at this microscope. So if you look into the microscope and you start seeing this, so what can you see? You can see a very thin layer, only one single layer of cell. So it must be one layer, it is simple flat cell squamous epithelium. So this is the simple squamous epithelium under the microscope. So it is only one thin film of cells. It is not going to be useful for protection, but it will be useful for diffusion, filtration. So we are going to use this type of simple squamous epithelium in the diffusion area, such as kidney or the lungs. At the end of the respiratory tract, we have alveoli. So these alveoli are lined by the simple squamous epithelium so that the oxygen we breathe in can go through this simple squamous epithelium and go into the capillary. And there will be gaseous exchange between oxygen and carbon dioxide. So that is the beauty of simple squamous epithelium. Although they are not useful for protection, they are useful for filtration and diffusion. So what about this? This is a square shape and cuboid shaped cell. This is the cuboidal epithelium. There is only one layer and therefore simple cuboidal epithelium. And because the cell size is bigger and therefore there is more things to do inside the cell, so they will be useful for secretion and absorption, like in our kidneys. Like the kidney will secrete bicarbonate and it will take up all the sodium and potassium back into the cell. And so this type of simple cuboidal epithelium will be useful for the secretion and absorption, and we use that type of epithelium in the kidneys. So look at this diagram. So the cells are tall. This must be the columnar epithelium. You can see the little cilia here. So it is ciliated. Is it one layer or multiple layer? It is not really a one layer, but it looks like a multiple layer. So it is a fake pseudo-stratified columnar epithelium with the cilia. So pseudo-stratified ciliated columnar epithelium. So this type of epithelium, we are going to use it in our trachea, which is the windpipe, which trap all the dust and filter the air to the best. Yeah, so that is the pseudo-stratified ciliated columnar epithelium. So what about this? This is a lot more simple, okay? Only one cell, but very tall, with one nuclei. It is not pseudo-stratified. It is simple, tall, columnar epithelium. And therefore, now we will use this type of epithelium in the gastrointestinal tract. Yeah, so in the gastrointestinal tract. So this is the simple columnar epithelium because the cell size is big. So there will be more functions they can do inside the cell. Right, so let's look at this slide again. So this is if you look at this picture, this is flat cells here, but there are more multiple here. So multiple layer of flat cell, that will be stratified squamous epithelium. The difference between this and that is we can see another layer, which is keratin layer. Keratin is a wax fibrous layer, which protects. So this is for the protective, very protective. You have wax on outside. On top of that, you have multiple layers, multiple layer of cells. And that is what we are going to use for our protection. This is for our skin. So our skin, if we look under the microscope, we will see something like this. Multiple layer of squamous cell, which is called stratified squamous epithelium, which is keratinized. So this page is a summary of what we have talked about. I want you to take your time, look at this diagram, this table, and try to absorb. So now we got to glands. What is a gland? Gland is, again, nothing more than a group of epithelial cells. As long as they can make some secretion, that is a gland. There are two types of glands in our body, which is exocrine gland and endocrine gland. What is the difference? The main difference is whether they have a deck or not. For example, endocrine gland, which is the thyroid gland, they do not have any decks. So what they do, they make thyroid hormone in their cell, and they put thyroid hormone directly into the bloodstream without any tax. So there is no tax. So what about the exocrine glands? These exocrine glands have tax, which mean, like, for example, like exocrine portion of the pancreas. Pancreas is both exocrine and endocrine gland. For the exocrine portion, they are going to make amylase, lipase. So these amylase and lipase, they will be formed by the cells, pancreatic cells, and they will be secreted into the intestine through the pancreatic duct. And therefore, they are exocrine glands. So just one little peculiar gland, which is only one cell, but which forms a gland, which is called unicellular exocrine gland. That is the goblet cells. They are usually found in our gastrointestinal tract. So now we come to the end of the epithelial cell. I want you to take your time, look at this slide, which is in our textbook, and try to appreciate what we have talked about. So now I'm trying to compare another slide here, which is very different from your epithelial cell slide. What is the difference? There is no polarity. So here's polarity. It is not acellular. There is no cellular. So this is the connective tissue. So let's go and study about connective tissue. There are 4 types of connective tissue. They are connective tissue proper, skeletal tissue, cartilage tissue, and blood. Don't forget, blood is our connective tissue. So this is the typical microscopic view of the connective tissue. You can see a cell here. You can see one cell here. You can see another cell here. You can see one cell here, but nothing to do with the epithelial tissue, which has multiple and lots of cells in epithelial tissue. This is not very cellular, and these cells are floating in the sea of matrix, which is little fibers here. So this is the matrix, and that is what we call connective tissue. So now you can appreciate what a connective tissue is under the microscope. So let's go and dig and zoom and high power microscope into this area. And this is what we see. You can see the matrix, which is made of fibers, like collagen fibers, elastic fibers, and reticular fibers. And you can see the cell here, fibroblast, lymphocyte, fat cell, mast cell, neutrophils, and the other space, they are filled with the gelatin-like ground substances, which are nothing more than glycoside, aminoglycan, proteoglycans. So the connective tissue is composed of cell matrix and ground substance. What is the characteristic of connective tissue? There are not many cells, small number of cells, large amount of matrix, and they will have ground substance. And their origin is mesenchymal origin. So microscopic features important is not many cells and large amount of matrix. That is the connective tissue. So when we talk about cells for the connective tissue, the cells can be fibroblast, chondroblast, or osteoblast. Fibro means fibrous tissue, blast means cell, fibrous forming cell, fibrous tissue, we get it. Chondroblast means chondro means cartilage, blast forming cells. Chondroblast, cartilage, yeah, we get cartilage. Osteoblast, osteo means bone, blast means cell, osteoblast, bone forming cell. On top of that, we have some fat cell, white cell, macrophage, and mast cell. So connective tissue matrix is made of collagen fibers, elastin fibers, and reticular fibers, which we have seen in our previous slide. And the rest area, we will just put it in the ground substance, proteoglycan, glycoside, aminoglycan. These are the substance, just to fill the gap and try to absorb water. So you might want to know, what is collagen made of? Collagen made of 3 amino acid chains spin together, that is collagen, elastin, like amino acid chain, reticular fibers, again, like amino acid chain. So this is the table in our textbook, which is again really good explanation of connective tissue, which can be connective tissue proper, cartilage, bone, and blood. And connective tissue proper can be subdivided into loose connective tissue and dense connective tissue. So comparison between connective tissue and epithelial tissue now it is very clear. Multicellular and not cellular at all. Polarity and there is no polarity, or big matrix and there is not much big matrix at all. So that's the difference. So now if you go to the connective tissue proper, it can be subdivided into loose connective tissue and dense connective tissue. Underneath the loose connective tissue, we will have areolar connective tissue, adipose tissue, which is adipose means fat tissue, and reticular tissue. And elastic tissue, dense regular collagens, they will go under the dense connective tissue. So let's look at this slide. If you look at this, and this is the photomicrograph. There are a few cells, but not as many. You can see a big matrix. Inside the matrix, there are many cells floating around. You will see this is connective tissue. This is the example of a areolar connective tissue, which is the tissue which supports the epithelium, lamina propria. So this tissue is connective tissue, and epithelial tissue sits on top of the connective tissue. So the connective tissue is a supporting structure of many other cells like epithelial cells or mast cells. So keep looking at that. And this is the photomicrograph of the adipose tissue. Big cells, nucleus are here and there, here and there. So this is not a simple squamous epithelium. This is the globules of fat. That is the adipose tissue, especially from the mammary gland. So if you look at that again, like, is it multicellular? Yeah, it is a little bit cellular if you want to argue like it is an epithelial tissue. But on the other hand, there is no polarity, and you can see the matrix and you can see the cells. So this is what we call the reticular connective tissue, which we can find in our spleen. Okay, so number 3 tissue is mast cell tissue. Again, like mast cell is composed of mast cell tissue plus connective tissue. So a mast cell is a composite tissue which is made of mast cell tissue and connective tissue. So there are 3 types of mast cells. They are striated, smooth, and cardiac mast cell. We can find striated mast cell in our skeleton, like our bicep mast cell or tricep mast cell, smooth mast cell in the gastrointestinal tract, and cardiac mast cell in our heart. So there are little difference between each and every mast cell. If you look at that, this mast cell, it is, you know that this is not a connective tissue. You know that this is not a epithelial tissue. And these cells they are running like a long tubular structures. They are running long tubular structures. And you can see the little striations here everywhere. Striations here is everywhere. They are long, cylindrical. They have multiple nuclei here and there, here and there. Multiple nuclei. And this is the example of striated mast cell. This is the example of the skeletal mast cell tissue. So this is a very good drawing or photomicrograph or whatever you say. This is a typical feature of striated mast cell. You can see the striations. You can see long and cylinder or tubular mast cell fibers running along each other. So this is the skeletal mast cell. So this is smooth mast cell. Smooth mast cell, there are sheath of flat cells, mast cells, piled on each other. Spindle-shaped cells with central nuclei. The nucleus are central, and it is very different from the mast cell tissue, striated mast cell, and we can find in the hollow viscous organs. So now we got to number 3, cardiac mast cell. it looks like they have striations. Yes, cardiac muscle is striated muscle, but the difference is they do branch. If you look at this muscle cell, they branch here and they branch here. They join here. So branch and join, branch and join. That is the typical feature of cardiac muscle. They look like striated. Yes, they are striated muscle, but they do branch and they do join. So this type of muscle we can see in our heart. That is the cardiac muscle. So now we understand skeletal muscle, smooth muscle, cardiac muscle under our microscope. So number 4 tissue is nervous tissue. And nervous tissue can be found in the nervous organ like brain and spinal cord and the periphery nerves. So a nervous tissue is made of neurons and supporting cell, which is what we call neuroglia cells. So this is a typical diagram of a neuron, which is like a jellyfish or starfish or octopus, multiple limbs going outside from the big cell body. This is a little bit more similar in what you see underneath the microscope. And this is the cell body. These are the dendrites. All the information from the periphery will be taken to the cell body. And cell body process this information and will be carried along the axon and go to the end of the axon where there will be secretory sites, something like acetylcholine will be secreted at the end of the axon. And there will be progression of the function or progression of the nerve conduction. So this is the typical diagram of the nerve neuron. So now you can see the difference between a nervous tissue where you need to see the neuron. You can see the muscles, like 3 types of muscle, nothing to do with the connective tissue or the epithelial tissue. Right. So neuron, cell body, dendrite, axon, they are supported by the neuroglia cells. What is a neuroglia cells? They are non-neuronal cells. They do not have nerve conduction capacity. Their job are to support the neurons and protect the neuron and form myelin. In other words, their job is to maintain the homeostasis, the internal liu, internal environment to maintain. So that is all about the lecture today. At the end, please remember 4 types of tissue in our body: epithelial tissue, connective tissue, muscle tissue, and nervous tissue.